Very good afternoon, everyone. So today I'm going to tell you about controlled drug delivery system. So controlled drug delivery system is one which delivers the drug at a predetermined rate for locally or systemically for a specified period of time. To get continuous oral delivery of drugs at predictable and reproducible kinetics for predetermined period throughout the course of GI. Controlled release drug delivery employs drug encapsulating devices from which therapeutic agents may be released at controlled rates for long period of time ranging from days to months. Such systems offer numerous advantages over traditional methods of drug delivery, including tailoring of drug release rates, protection of fragile drugs, and increased patient comfort and compliance. Controlled drug delivery system can increase the maintenance of drug level within a desired range, the need for fewer administration, optimal use of the drug in question, and increased patient compliance. Now, the history of controlled drug delivery system. The science of controlled release was first originated from the development of oral sustained release products in 1940s and early 1950s. The first patent for oral sustained release preparation went in favor of lipovis. This preparation contains small coated beads that were releasing the drug slowly and constantly. This idea later developed by Whiteley and launched the first marketed sustained release products in 1950. Over the past 30 years, as the complication involved in the marketing of new drugs increased and various advantages recognized of controlled drug release, the greater attention is being paid in this. Now, what are the ideal characteristics of this controlled drug delivery system? First of all, it should be inert, it should be biocompatible, it should be mechanically strong, it should be comfortable for the patient, it should be capable of achieving high drug loading, it should be safe, it should be simple to administer and remove, and it should be easy to fabricate and sterilize. Now, there are various terminologies and definitions which come under controlled drug delivery system. So, according to United States Pharmacopoeia, the modified release dosage form is the one for which the drug release characteristics of time course or the location are chosen accomplish therapeutic or convenience objectives not offered by conventional dosage forms such as solutions, ointments, and dissolving dosage forms. One class of modified dosage form is extended release dosage form, which is defined as the one that allows at least twofold reduction in dosing frequency or significant increase in patient compliance or therapeutic performance when compared with the presented as a conventional dosage forms, which are the solutions or the tablets. The term controlled release, prolonged release, and sustained release and long-lasting release had been used as a synonyms with the extended release. Controlled drug delivery system is one which delivers the drug at a predetermined rate for locally or systemically for a specified period of time. And sustained drug release allows delivery of specific drug at a programmed rate that leads to the drug delivery for a prolonged period of time. Prolonged release products release the active ingredients slowly and work for a longer time. It is a dose of medication over an extended period of time. Now, the difference between controlled release and sustained. Controlled drug delivery is one which delivers the drug at a predetermined rate for a specified period of time. The controlled release is perfectly zero order release, that is the drug release over the time irrespective of concentration. And the sustained release is defined as the type of dosage form in which a portion of the drug is released immediately in order to achieve a desired therapeutic response more promptly. And the remaining, the maintenance dose is then released slowly and thereby achieving a therapeutic level which is prolonged but not maintained constantly. The sustained release implies slow release of the drug over a specified period of time. It may or may not control release. Now, what is the rationale of controlled drug delivery system? Its utility is to maximize the reduction in the side effects, cure or control the disease, administer by a suitable, immediate release of the delivery, achieve prolonged therapeutic effect in drug release kinetics, and deliver the drug at a predetermined rate locally or systemically. Now, what are the advantages of controlled drug delivery system? to improve the patient compliance, to reduce the fluctuation in the steady state level, to increase the safety margin of high potency drugs, reduction of total health care costs, reduction in the frequency of drug administration, improved patient compliance, reduction in the total drug uses when compared with conventional therapy, reduction in drug accumulation with chronic therapy, reduction in drug toxicity, stabilization of medical conditions, improvement of bioavailability of some drugs because of spatial control, economic to the healthcare providers and also to the patient. Now, there are several industrial or the commercial advantages, like illustration of innovative or technological leadership, product life cycle extension, product differentiation, market expansion, and patient exchange. There are some disadvantages to control drug delivery system. They are that it decreases the systematic availability, poor in vitro in vivo correlation, increased risk of toxicity, retrieval of drug is difficult in case of toxicity or poisoning, the possibility of toxicity or non-biocompatibility of the material goods, undesirable byproduct of degradation, any surgery required to implant or remove the system. The chances of patient discomfort is more. The cost of the product is high. Delay in the onset of drug action is also there. Sometimes there is dose jumping also. And there is sometimes an increase in first pass metabolism also. There is a greater dependence on the GI residence time of the doses form. Possibility of less accurate doses adjustment in some cases. Cost per unit dose is high when compared to conventional doses. Not all the drugs are suitable for formulating in this controlled drug delivery system. Now, there are some characteristics which are required for selection of drug candidates, like for formulating any formulation into controlled drug delivery system, the drug's molecular weight or size should be less than 1000. Solubility should be greater than 0.1 microgram per ml for pH 1 to pH 7.8. The pKa value should be greater than 0.1% at pH 1 to pH 7.8. The apparent partition coefficient should be high. The absorption mechanism should be diffusion. The general absorbability should be from all GI segments, and the release should not be influenced by pH or the enzymes. Now, there are some pharmacokinetic parameters also, like the elimination half-life should be between 2 to 6 hours. The elimination rate constant should be as per the design. The total clearance should be dose independent. The intrinsic absorption rate should be greater than the release rate. The apparent volume of distribution should not affect the required amount of the drug. The absolute bioavailability should be greater than 75% or more. The steady state concentration should be lower than CSS and smaller than B. And the toxic concentration should be, the therapeutic window should be broad. Now, there are also some other characteristics, like it should have a shorter elimination life or very longer elimination life. It should have a narrow therapeutic index, poor absorption or very active absorption, low or slow absorption, and extensive first pass effects. Now, there are some approaches to design the controlled release formulation. Like first is the diffusion. And diffusion comes under two categories: reservoir type devices and matrix type devices. Next is the dissolution. Dissolution contains encapsulation, seed coated, microencapsulation, and matrix release. Next is ion exchange principle. Now, first of all, we are studying about diffusion. Now, diffusion, what is this? It is the movement of molecules from a region of higher concentration to a region of lower concentration down the concentration gradient. Drug diffusion is very much essential mechanism for drug dispersion throughout the biological system. Now, first of all, we will study about the reservoir diffusion system. It is also called as laminated matrix devices. It is a hollow system which contains of an inner core which is surrounded by a water-insoluble membrane, and a polymer can be applied by coating or microencapsulation. The rate controlling mechanism is that the drug with the partition into the membrane and the exchange with the fluid surrounding the drug by diffusion. Commonly used polymers are HPC, cellulose, or polyvinyl acetate. Now, the next is the matrix dissolution system. The matrix dissolution system is again of two types: the rigid matrix diffusion and the soluble matrix diffusion. The rigid matrix diffusion materials used are insoluble plastics such as PVC or fatty acids. The soluble matrix system, it is called as glassy hydrogels and popular for sustaining the release of highly water-soluble drugs. Now, the next is dissolution. Dissolution is the process by which a substance forms in the solution. When administered orally, a drug has to first dissolve in the GIT and then before it can be absorbed in the blood and reach the site of action. Now, dissolution, this is the whole flowchart diagram of the dissolution process. Like you can see the polymer membrane with different dissolution rates. At time 0, at time t, the drug is released, and you can see the matrix dissolution control and undissolved matrix, the difference between the two. Now, encapsulated dissolution system. This is also known as the coating dissolution control system. Dissolution rate of coat depends mainly upon the stability and thickness of the coating. It marks the color, odor, taste, and minimize the GI irritation. The controlled release products, by decreasing the dissolution rate of the drug, which are highly water soluble, can be formulated by preparing appropriate salt or derivatives, by coating the drug with a slowly dissolving material, or by incorporating the drug into a slowly dissolving gas. Next is the matrix dissolution system. It is also known as monolithic dissolution control system. The dissolution is controlled by altering the porosity of the tablets, by decreasing its wettability, or by dissolving at a slower rate. It follows the first order drug release. The drug release can be determined by dissolution rate following. Now, the next comes the ion exchange. Ion exchange is a reversible interchange of one kind of the ion present in the insoluble solid with another of the like charges present in the solution surrounded by the solid, with the reaction being used, especially for softening or making the water demineralized, purification of chemicals, or the separation of the substance. Ion exchange resins. They are called the cross-linked water-insoluble polymers, which contains ionizable functional groups. These resins are used for taste masking and controlled release systems. The formulations are developed by embedding the drug molecule in the ion exchange resin matrix, and this core is then coated with a semi-permeable membrane, which can be ethyl cellulose. This system reduces the degradation of drug in GIT. The most widely used and safe ion exchange resin is divinyl benzyl sulfonate. In tablet formulation, ion exchange resins have been used as a disintegrant, and they are used in the manufacturing of pharmaceuticals, not only for catalyzing certain reactions, but also for isolating and purification of pharmaceutical active ingredients. Three ion exchange resins are mainly used: sodium polystyrene sulfonate, polystyrene, and polystyrene. Sodium polystyrene sulfonate is a strong acidic ion exchange resin which is used to treat hyperkalemia. Polystyrene is a weakly basic ion exchange resin, and it is used for treating of hypercholesterolemia. And the last one is strongly basic ion exchange resin, and it's used to treat hypercholesterolemia. And cholesterol and cholesteremia are commonly used as bile acid